mediated endocytosis pathway. The idea is there's one endosome that's being endocytosed, and you can look at other fluorescently tagged proteins in conjunction with clathrin. And when clathrin reaches a maximum, that's considered the maximum size of your endosome. That's actually when it's become mature, and that is going to separate from the actual plasma membrane. And so what you do is you look at the fluorescence of the other proteins of interest and look at them relative in time to clathrin. And then you can tell, is it at an early phase of clathrin-mediated endocytosis or a late stage? Just by basically where the trace peaks relative to clathrin up there. And so one thing they noticed with this particular bar domain protein is that it peaked pretty much where clathrin does. And so that was quite interesting because other proteins like dynamin come a little bit later than clathrin, and same with the RT3 complex or actin assembly. So they come after where this protein does. So it kind of suggests that this bar domain protein is coming on first before the arrival of actin assembly and the proteins that are needed to actually size precision inside this endocytic invagination. Another approach that people use to look at where this protein is in relationship spatially relative to the plasma membrane is to use a combination of turf microscopy with wide field microscopy. So in turf microscopy, you're limited to a very finite focal plane just on the outside of the plasma membrane, whereas wide field you can see far into the cell. So if you look at the relationship of where a protein disappears Okay, so if you look at basically the difference here, so if a protein is right at the plasma membrane, it's going to start disappearing away from when it starts moving up into the wide field. That means it's moving away from the plasma membrane and moving along the invagination of endocytosis. And so, for example, if you were to look at AP2, the plasma adapter protein, that's exactly what you would see. Over time, it starts moving away from the plasma membrane. But with this particular flat bar domain protein, it stays right along wide field and starts to see the same thing at the same time. So that means it's always at the plasma membrane. Furthermore, using super resolution approaches, they could actually see that there was a separation. That distinctly that this protein was at the plasma membrane, whereas clathrin was forming a cage that was separated from that. So that's pretty amazing because that membrane that is thought to be next to the clathrin, the endocytic invagination, is thought to be flat. And that was fairly unusual. Bar domains weren't typically associated with flat membranes in the past. And so they wanted to know what this thing was doing. And so another assay that we commonly look at with bar domain proteins is that they frequently like to activate the actin cytoskeleton. And so if you were to look at this assay using NWASP, like I said before, NWASP is auto-inhibited, and so therefore it shouldn't be active underneath normal conditions, but if you just take the C-terminal tail of it, it is active. And that's what we would see here. So if you mix the C-terminal tail with our RT3 complex and actin monomers, you differentiate actin polymerization. But if you were to take the full-length protein, mix it with RT3 complex and actin, you would not see actin assembly. However, if you could bring a protein that could activate NWASP, like this bar domain protein, then you should see actin assembly if you could activate this protein. And it turns out this protein loves to bind to PI3-4P2 or PI4-5P2 phosphonosinucleotides that are found in lysosome. And when it does that, it actually markedly enhances actin assembly. So that says this protein binds to flat membranes, binds to NWASP, activates NWASP at flat membranes. And furthermore, it was actually shown to actually bind to larger vesicles. So it preferred, rather than small vesicles like regular bar domains do, this prefers much bigger ones, like a flat bar domain. So it's consistent with that. So they came up with this model where this protein was actually binding at the plasma membrane, bringing in NWASP, which was helping to drive actin assembly along the clathrin invagination. So this was a pretty big deal because for a long time we knew actin was around the endocytic invagination. But it was kind of perplexing because you think about the forces that are happening here. First, we're pulling on the membrane. And what I said before is that there's always a restoring force. When you pull on the membrane wants to snap back to its original position. So you always have that. But in this model, with the actin polymerization, the bar that's pointing up here, it would imply that actin assembly is actually in the direction of the imagination. So it would seem like actin assembly here is useless. You're actually making things worse. You're not actually helping to make a growing endosome. But with the advent of this bar domain protein, now we can form this coat up here, and it stabilizes that. It prevents that membrane from bending. So now you're pushing on that stabilized membrane, which is naturally going to cause the entire actin network to flow downward. And if you can grab onto that flowing actin network with certain adapters, in which there are in these cases, SLA2 and M20, we're not going to discuss them, but they just bind clathrin and this actin, then basically you can just hang on to the actin while you just write it down. But there's still a problem here. We still haven't balanced our forces here. So what's contributing to the rest of the situation? The answer is a whole lot of bar domains. There's a lot more bar domains that actually bind to this thing because there's a whole mess of curvature that's happening during clathrin-mediated endocytosis, where you're pulling this thing from the membrane, you create all sorts of interesting geometries for bar domains to bind to, and the more you pull, the more geometries that you have. And so, as a consequence, they found that there's a bunch of bar domains that wind up binding in a very sequential way along, first of all, you have this FBP17 and starting next to 9 that bind around at the base of this thing, and then you get endothelin amplification binding as you begin to pull on this neck region. And in fact, there's a structure of endothelin bound to a lipid tubule, and you can see that it forms this nice helical array with this really equal diameter all the way across. Interestingly, the spacing between these rungs where the bar domain lie is enough to fit in a key domain for dynamin, as well as another protein called synaptogenin. Synaptogenin is important for the disassembly of the clathrin coat later on. But this is a key for the binding of dynamin, which is key protein for severing endotubules. Okay, so how do we sever an endotubule? Well, we need, like I said, dynamin. Dynamin's a GTPase, that oligomerizase. And we need that. We also need bar domain protein because we need to stabilize that membrane curvature. And then we also need some actin polymerization to happen. And so that was known because if we take an F-bar protein and we overexpress it inside the cells, what we see is a bunch of tubules. You can see these big lines here. It's not very clear in here. But they happen to co-localize with actin, so you see these tubules that are here. But if you co-express this with dynamin, what you see is just a bunch of dots. You don't have any more tubules. So that suggests that the dynamin is severing these tubules. But this is somewhat interesting because if you were to look at this in vitro, you could see this F-bar protein makes these tubules, dynamin also makes these tubules, and if you put them together, you also see tubules. So that was kind of perplexing because that doesn't imply severing. So the answer was actin. So if you were to treat your cells with latrunculin B, which latrunculin B sequesters actin monomers. So that means you're not going to get actin polymerization. So you stop actin polymerization. Then you get all these tubules forming. But if you were to wash it out, then you start seeing severing of these tubules. You can literally see that here's a nice straight tubule. Over time, you see it becoming perforated. So you need actin assembly, bar domains, and dynamin to sever tubules. And that's true everywhere. So, dynamin. Dynamin is this, it's a dimeric GTPase that can oligomerize through its stop domain, but it can also oligomerize through the GTPase domain itself. And as a consequence, it can form these large helical arrays, and this helical array can actually change its diameter in a GTP-dependent manner. So as it goes through its GTPase cycle, it works as a ratchet, where the heads start like moving in, putting tension on the membrane, and they twist. And so for every hydrolytic cycle, they twist one step. And so by doing that, they ratchet on this membrane neck to pinch it up. And so dynamin's been implicated in a lot of membrane fissioning events, but it was only recently that they actually figured out how or where the membrane gets severed relative to dynamin. Seems convenient to just say, okay, you're going to ratchet, but eventually just make a narrower and narrower diameter. At some point, you have to like literally sever the membrane. But no one really knew how that actually happened. And so there's a common assay that we use to actually study membrane forces on membranes, and this uses an approach called venomaspiration. So you just take a little micropipette aspirator and you suck on GUV. And so now that GUV will be stuck underneath the suction. Then you can take an optical trap and just pull on the membrane. And you can pull little tubules up on the membrane. And so this is kind of what it looks like. And then eventually, you can keep pulling on that membrane and eventually it severs. And it turns out where it severs is actually at the junctions between dynamic zones and where there is no dynamic. So it always severs at the barrier of dynamic. And which is just illustrated with another example here. Now, endothelin is quite interesting. So if you add bartamate to this exact same assay, what you'll notice is that the force goes to 0. So without endothelin, and you're pulling on this membrane, you can feel this large tension on the membrane. But like I said before, bartamate reduces that membrane tension. In fact, it causes it to go to 0 in this case. And it requires a huge amount of force, over 50 piconewtons, which is much more, many times greater than that generated by like a myosin 2 or something like that, in order to actually sever those tubules. Which is quite intriguing because if you add this in conjunction with dynamic, what you see is that in 8 seconds, this is the same force in both cases here, but under 8 seconds without endothelin, the tubule snaps, but with endothelin, it persists. And in fact, they actually co-assemble onto these same tubules. And this is actually, this kind of mechanism of using endothelin and dynamic and active assembly works in all membrane severing events. So including that in non-platinum mediated approaches. So, for example, there are these pathogens that secrete this thing called shigatoxin. And shigatoxin, basically, once it gets into the cells, it shuts down protein synthesis. And so it actually enters through a pathway that isn't quite clear. It somehow forces the actin cytoskeleton to pull in where the shigatoxin is. And so when it does, it creates these large tubules that you can see up here. And these large tubules actually overlay with actin. But you can also see dynamic localizing to these tubules at these very specific junctions. And you can also see endothelin also localizing to them. So this just illustrates dots where they're localizing with the shigatoxin, and then this is endothelin. And it turns out that the severing efficiency is markedly enhanced when you have dynamic, actin, and endothelin together. But if you were to knock out all of these proteins, you would stop severing altogether. Okay. So, disassembly of platinum. So after you've now successfully pulled away the membrane and you cut it off using the dynamic system, you have this membrane vesicle entombed by platinum. So now you got to get rid of it. So it turns out that there are 2 proteins that are really important for this. One is synaptogenin, which actually is dephosphorylating this PI345P3 to PI34P2. And that's important because the next protein that comes in there, called auxilin, will only interact with platelin underneath that condition. And in fact, there are mutations in synaptogenin that contribute to Parkinson's disease because they basically shut down the processing of factor-mediated endocytosis. So once auxilin is present there, it recruits these chaperones. So the HSC70 is a common chaperone. And then it turns out it has to recruit 3 of them at a time, and when it does that, it can rip off one triskelion from the surface of the membrane. And so that process would just repeat iteratively until you remove the entire triskelion. And it was shown that if you were to look at neurons of an auxilin knockout mouse, you would actually see that these neurons would have a ton of plaquenil spots inside of them when compared to control, but the same is true with synaptogenin. So indicating that it's necessary that we have both of these proteins at play. Okay, so once we have an endosome, now we got our endosome, we stripped away plaquenil, now we have proteins inside of it that we want to keep and other proteins that we want to get rid of. So now that enters in a system called the recycling endosome. So here, there's going to be a decision made as to whether the protein of interest is going to be kept or sent down the lysosomal degradation pathway. And so this is a lot of this has been worked out for a couple of common receptors like transferrin receptor, NCI, MPR. And these are commonly used assays in the lab when you're looking at recycling. because you can basically take an antibody. These are cell surface expressed protein that they'll be found at the cell surface. You can add an antibody that marks